Good afternoon, everyone, and welcome to the University of Manitoba Alumni and Friends Virtual Learning for Life program. My name is Tracy Bowman. I'm the Director of Alumni Relations here at the University of Manitoba and a very proud alumna. Before we begin, I'd like to acknowledge that the University of Manitoba campuses are located on the original lands of the Anishinaabe, Cree, Oji-Cree, Dakota, and Dene peoples, and on the homeland of the Métis Nation. We respect the treaties that were made on these territories. We acknowledge the harms and mistakes of the past, and we dedicate ourselves to move forward in partnership with Indigenous communities in a spirit of reconciliation and collaboration. So welcome to today's presentation. This is our third session of our Spring 2021 series. I appreciate you all making this part of your day, whether you're here live or you're watching it later on after today. This is our fourth Virtual Learning for Life series that we've been able to offer since last May, since the start of the global pandemic. We've seen great interest in this program, and we're so glad that you're joining us, whether this is your first lecture or your 27th. All previous lectures live on our website, so please go to the alumni section of the umanitoba.ca website and you can look under Virtual Learning for Life, and all of our sessions are there from as way back as last May. So there's lots of great content, lots of amazing speakers. I really encourage you to check some of those out at another time. We are able to offer this program free of charge to our alumni and friends thanks to the very generous sponsorship of our affinity partner, IA Financial Group. Many thanks to them. You can learn more about their insurance options if you're interested that they offer to alumni on the alumni website. So before we get started, just a few housekeeping details. As you see, we're recording today's session. That's the only way you'll be able to view it again. We'll send you a link after today's presentation, as well as I said, it lives on our website. Please watch this many times, share with friends and family, lots of great content. Our format is fairly typical, where our speaker will present for about 35-40 minutes. Then we will open it up to questions afterwards. If this is, you're returning to the Virtual Learning for Life program, you know that we use a platform called Slido.com. This is how we are able to get questions from you. So if you want to go to Slido.com, and the passcode is VLFL19. And I encourage you to bring that up on your internet browser right away. So if a question pops in your mind when our speaker is presenting, you're able to ask those questions. So I'll continue to monitor that, and we'll try to get through as many questions as we can before the end of the presentation. So now I'm delighted to introduce today's speaker, Dr. Soheila Karimi, who will be speaking on research advances in pathogenesis and therapeutic development for multiple sclerosis. So let me tell you a little bit about her before she starts. Dr. Karimi is a full professor and a neuroscientist in the Department of Physiology and Pathophysiology in UM's Rady Faculty of Health Sciences, and is also a scientist with the Children's Hospital Research Institute of Manitoba. Dr. Karimi directs an internationally recognized research program in neural regeneration and stem cell research with a focus on a treatment development for spinal cord injury and MS, aiming to improve the quality of life for individuals affected by these very debilitating conditions. Her pioneering work in cellular therapies has been a landmark in this field and has contributed to the launch of the current stem cell clinical trials for individuals with spinal cord injuries. Her research on MS has also discovered new treatment targets and potential disease biomarkers. Her program has been supported by the continuous generous support of nearly $8 million in research funds from national and international sources. And in 2020, Dr. Karimi was named one of Canada's top 100 most powerful women by the Women Executive Network and received a Manulife Award in Science and Technology. Wow. So with that, over to you, Dr. Karimi.
Okay, thank you so much, Tracy, for the kind introduction, and good afternoon, everyone. It is my absolute pleasure to speak to you about some of the research advances that we have made in understanding the pathogenesis of multiple sclerosis and also therapeutic development for this disease. And it is timely, basically, to give this presentation because May is the Multiple Sclerosis Awareness Month in Canada. And when we look at multiple sclerosis, so as like introduction to this disease, this is an immune-mediated demyelination disease of the central nervous system. So let me just see. So when we look at CNS or central nervous system, it contains brain and spinal cord. And within the CNS, we have nerve fibers that are running throughout the body, and then they are basically controlling our function. These axons or nerve fibers are ensheathed with a fatty structure which is called myelin sheath, and this myelin sheath is the target in multiple sclerosis. Basically, through immune-mediated attacks, this myelin is destroyed, and we see patches or plaques of demyelination within the brain and spinal cord. MS was basically defined and named in 1868 by John Martin Charcot, who was a French neurologist. And basically, the term multiple sclerosis started from that point. When we look at the epidemiology of MS, so MS is known as Canada's disease because we are home to one of the highest prevalence of MS in the world. And also in the prairies, including Manitoba, we have the highest prevalence within Canada. Currently, there are over 90,000 Canadians living with MS, and around the world, we have 2.8 million individuals affected by MS. Each day in Canada, 12 individuals are diagnosed with MS. And as we talked, so Canada is a center basically for MS because of the overall number of individuals who live with MS in Canada. And the other fact about MS is that it's affecting mainly younger adults, and it manifests sometime between age 15 to 40. And as mentioned, we have the highest prevalence of MS in the world, 291, based on basically the last statistic in every 100,000 people. When we look at kind of pathogenesis of MS, currently we don't really know how MS is initiated. This is a question that we are all working on, that research groups around the world. But some facts that we know is that there are some predisposing factors that trigger MS, including genetic factors. For example, there are 200 genes that they have been associated with MS. Also, it seems that there is genetic component that make a person susceptible to MS, because there is 30% chance between identical twins to have MS, and 2 to 5% if a sibling has MS. There is a sex bias because there are 3 times more female affected by MS than male. Also, MS can be initiated by some viral infections. Pollutants and smoking definitely increases the progression rate of MS. Also, recently, it has been shown that excessive salt intake can also trigger MS. And one of the really prominent kind of, predisposing factors at the moment is vitamin D deficiency. When we look at vitamin D deficiency, it's now known as a risk factor for MS, and because the prevalence of MS is also increased in northern climates with colder temperature like Manitoba, where vitamin D deficiency is also common. And what we know at the cellular and molecular level is that when vitamin deficiency is at a healthy level, this can basically generate a balanced, healthy immune response. And when vitamin deficiency happens, this balanced immune response is disrupted, that trigger MS. So this is one of the treatment strategy. If MS individual shows vitamin deficiency. So this is one of the current treatment strategy. In terms of symptoms of MS, MS is a very heterogeneous and diverse disease because it can happen in the brain. It can affect optic nerves, which would then affect visual function. It can also affect the spinal cord. So depending on where the plaques of demyelination or MS lesions are found, so there are also different symptoms. But the leading symptoms are numbness, tingling, fatigue, muscle spasm, pain, and walking difficulties, and followed by other symptoms. So this shows basically the impact of MS, and also in terms of in Canada, with the number of MS patients, this actually shows a high socioeconomic, basically, area that needs to be addressed. When we look at MS diagnosis, so at the moment there are various tests that are done to basically identify if the symptoms are MS or not. And these are including blood tests or a spinal tap to get the cerebrospinal fluid through lumbar puncture. And this is normally to check any abnormalities at the level of antibodies or even some blood markers to rule out infections and other conditions like other autoimmune conditions that they have symptoms similar to MS. But these blood tests or spinal tap, they are complemented by some neurological testing to see how healthy is brain and spinal cord, by looking at basically visual function or by looking at the physiological properties of brain and spinal cord in legs and arms to confirm if there is any neurological impairment or not. And of course, magnetic resonance imaging or MRI is one of the very useful tests to check MS lesions in the brain and spinal cord. For example, this is MRI from MS individual, this is from the brain, and this is from a spinal cord. All the areas that are identified by arrowhead, and you see kind of, lighter area. These are demyelinating plaque, which shows MS lesions, and the same thing with the spinal cord. So to talk about a little bit how basically CNS is affected by MS. So I'm just talking about the structure of brain and the spinal cord. So again, this is brain, this is the spinal cord, and we have nerve fibers that are running through brain and spinal cord, and also controlling the rest of the body in terms of our body function. Receiving sensory information and also generating motor and locomotion, and also other functions in the body. So central nervous system controls our body function. When we look at the cellular molecular levels, so these cells that you see here, the big cells, they are called neurons or nerve cells. And they extend fibers, which are called axons. And these axons are ensheathed by myelin that we just talked about. So fatty kind of layer structure that are wrapped around these neurons. And myelin ensures rapid signal transduction through CNS. So the ability to touch a hot plate and very quickly, within milliseconds, even second, so just remove our hand from kind of hot plate is because of the rapid signal transduction between sensory and motor pathways in our body and CNS to basically make us aware of the harmful situation. And this is basically facilitated by myelin. Myelin are formed by cells, by the type of cells which are called oligodendrocytes. And as we talk in MS, myelin is attacked by immune cells, and then subsequently also oligodendrocytes die because of loss of myelin. Okay, so we just talked about immune attack. We don't know how immune response is initiated in MS against myelin, but what we know is that if in blood, in basically our immune organs, there are immune cells that are overactivated and they enter the bloodstream. And then through blood, these overactivated immune cells, which we call them leukocytes, they are now entering CNS tissue. Under normal circumstances, CNS tissue is completely separated from blood by blood-brain barrier. This is a structure that protects our brain and spinal cord from toxins, from immune cells, from harmful, basically, material. But then in MS, somehow this blood-brain barrier is disrupted, and then overactivated immune cells, they find their way to the CNS tissue. And these cells are T cells, T lymphocytes, B lymphocytes, macrophages. They start attacking myelin and basically destroying that. And this is through autoimmune. So basically patient's own immune cells overactivated, attacking its own myelin. And that results in MS inflammatory lesions. So this is CNS. This is a blood vessel at the center. And these are all infiltrating leukocytes within CNS tissue. And this is how this lesion expands. And within this lesion, then myelin is destroyed. So we talked about myelination and oligodendrocytes who are normally making myelin, but because of immune attack, these oligodendrocytes are dead because myelin is basically destroyed. And now we have axons or nerve fibers that they do not have myelin. And these demyelinated axons actually are the demyelination plaques, MS plaques, that you also saw on MRI images. So if these axons are not remyelinated, they eventually degenerate. But the good thing is that, especially at the beginning of MS, we have episodes of remyelination. So when myelin is destroyed, body tries to make new myelin. So there are different clinical subtypes of MS, which I'm going to very briefly describe them. So one form that is the most common is relapsing remitting MS or RRMS. So in this subclinical type, we see period of relapses. When we say relapses, is immune attack and demyelination. So basically, CNS, the area that attack is happening, so is losing its myelin and oligodendrocytes. Then this period of relapses are followed by remission. So the body try to heal. So brain and spinal cord try to regenerate new oligodendrocytes and remyelinate those axons that they have lost the myelin. And this is the period of kind of remission. And then again, another attack happens and another after one. So these are unpredictable attacks. The other form of MS is primary progressive, is that at the onset when MS happens and diagnosed and starts, it gradually progresses without relapsing remission pattern. So it's kind of progressive type. And degeneration of nervous tissue and demyelination gradually happens. The other form is that at the beginning is like relapsing remitting, but after some period of time, it becomes progressive. And this is secondary progressive MS. Another one is progressive relapsing. So it is progressing, but still we see relapses. Okay, so something that now I put kind of, these subclinical forms to describe how these episodes of MS actually happening, and then when inflammation happens, when degeneration happens. So these are really common subclinical points that we talked about them. In addition to that, I want to introduce another kind of subtype of MS, which is called clinically isolated syndrome. This is when the individual has one MS-like episode of demyelination, but then that's just the first episode. And they may or may not convert to relapsing remitting. So this is basically initial kind of a stage of MS. At the beginning, in all of these subclinical types, so we have more relapses, which actually shows that inflammation is predominant. But then as MS progresses, it becomes less inflammatory and it becomes more neurodegenerative type of disease. And this is a period that even though that inflammation is not happening that much and is reduced, nervous tissue has lost some of the basically cells and it progressively degenerate. And this is the basically area that we would like to curb the progress. So basically bring down the progression of neurodegeneration in MS individuals. There are a number of disease-modifying treatments at the moment for MS, and this is a relatively kind of, recent, basically, discovery and advances. The first kind of, approved drug for MS, it was in 1993 with interferon beta. I'm not going to go through any of these disease drugs because all the information is available out there. But if you look at the evolution of drug development, so you see that a number of drugs have been generated. But the majority of these drugs, they are actually working at the inflammatory and immune response, basically trying to slow down inflammation and trying to slow down immune attacks. At the moment, none of these drugs are helping with the regeneration aspect of MS to basically curb neurodegeneration and progression of MS. And this is the area that it's major gap in our basically treatment development. Something that is very important for MS individuals is that they receive the treatment as early as possible, because research has shown, clinical research has shown that if a patient receives treatment, disease-modifying treatment at the symptom onset, that would really then curb kind of progression and disability over time. And then if later treatment actually is exercised, so then that is definitely with increased disability. So the sooner, the better. And if MS remains untreated and just follow the natural course of MS without any treatment, so definitely disability arises. So that is the current basically, clinical management of MS, that the patient is recommended to receive disease-modifying treatment at the earliest stage and stick to that. So what happens if MS remains untreated? So what we talked was about demyelination, loss of myelin because of immune attack. But we also talked about the process of remyelination, which is basically new myelin-forming cells or oligodendrocytes are regenerated within our brain and spinal cord, and they ensheath and basically renew the lost myelin sheath around the axons. And this is where remission happens. When our when the body has the ability to regenerate oligodendrocytes and myelin, so patient they can have basically remission period. But if remyelination fails, which is normally at the progressive stage, then these nerve fibers that they stay demyelinated for such a long time, they degenerate permanently and they are gone. And when nerve fibers are gone, so it's basically results in permanent disability. So what we are trying to do in our research group is that to understand why remyelination fails and try to promote remyelination during the progressive phase of MS to be able to basically curb neurodegeneration. As I mentioned, a significant challenge in MS is really progressive MS and neurodegeneration. And this is the area that it has been slow advances, but research is undergoing, including research in our own lab. And as I mentioned, so progressive MS means that we lose axons. So this is healthy spinal cord. All the dots that you see, these are cross sections of nerve fibers, packed nerve fibers. But then look at MS spinal cord, many of these nerve fibers are gone. And this is where disability arises. And locomotion and mobility basically is reduced. And disability, as I mentioned, is increasing. And there is also end organ damage in MS, and even in progressive MS and untreated MS, severe forms, we even see that brain is basically shrunk. So what we are working at the moment, and this is the area of gap, is that first of all, understand how pathogenesis of MS is happening, basically why these immune attacks are initiated, and then also understand why the process of remyelination and tissue healing is slow in progressive MS. Understanding these concepts, that would allow us then to have targeted treatment strategy for progressive MS, and also identify disease-specific treatments based on the pathogenesis and pathology of MS. Okay, so one thing that you have been hearing, as I go through my talk, is that we need to regenerate new oligodendrocytes in MS because these are the cells that are targeted. And these cells, when they are regenerated, so they can then remyelinate axons. But when we look at the process of oligodendrocyte regeneration and remyelination, this is a tightly regulated process, cellular process, that oligodendrocytes start from either oligodendrocyte precursor cells or a population of neural stem cells within the brain and the spinal cord. These are cells that are immature, but they have the potential to become mature myelinating oligodendrocytes. But to get to that point, they have to go through lots of stages. They have to mature, start, extending their processes, find axons, and then wrap myelin. And what we have been trying to do in our research lab is that to understand how this process happens and what factors are needed for remyelination and regeneration of oligodendrocytes. For this, we have generated experimental models of MS. And one of them, which is a gold standard animal model in MS, is called experimental autoimmune encephalomyelitis, or EAE. And we try to basically mimic the pathogenesis of MS as much as we know by basically vaccinating mice with myelin peptides. And these myelin peptides, when they enter the body, So then immune system starts making antibody against these myelin peptides. And then when the level of antibody against the myelin is increasing, then we see the onset of disease and symptoms. We see demyelination. And in this mouse model, the peak of the disease around day 16, and then they recover, like what we see in MS individuals, partial recovery. And then we also, when we look at kind of the lesion, we can produce similar lesion in MS, also in EAE. And the way that this model works is that, as I mentioned, so we are eliciting immune response against myelin, and then all these peripheral immune cells, which are exposed to myelin peptides, they are overactivated, start making antibodies, and then also they find their way to the central nervous system, as we talked. They enter brain and spinal cord tissue, and they start damaging myelin. So I'm going to share now one of our very recent study that published last December 2020, that we have identified an endogenous target with the use of these MS animal model and also some MS patient samples, that we are showing that a protein which is called neuroglin 1 beta 1, it seems to be implicated in pathogenesis of MS, and also it has some regenerative capacity, and we think that this is a targeted treatment that can not only modulate the immune response, but it can help with regeneration of myelin. And I'm going to show you some evidence of that. So first of all, what we found was that in the EAE mouse, that in the area that myelin is destroyed compared to the healthy area, we also see a very significant decline in the level of NRG1 protein. So when we saw this, we asked that, okay, so what now this protein is declined, is depleted in MS, and we found that actually it's depleted very early during the EAE kind of evolution in the mouse. So the next thing that we asked was that, okay, if we start treating the mice, the EAE mice, with this peptide, what happens? And what we found was that in mice that are treated with NRG1, we have less leukocyte infiltration into the CNS. All the green signal that you see in the EAE mice, these are all infiltrated leukocyte. Why in healthy kind of regions we don't have immune cells? Because as I mentioned, because of the blood-brain barrier or blood-spinal barrier, immune cells, they cannot enter the CNS tissue under normal circumstances. But with EAE or MS, we see abundant number of leukocytes. And these are initiating basically demyelination and destroying the tissue where they are. And then with NRG1 treatment, we were able to see way less number of leukocytes, not only in the spinal cord, but also in the blood, which was very promising. And normally these leukocytes, when they enter CNS tissue, they start releasing lots of cytokines, which are called pro-inflammatory cytokines, that are really initiating the process of tissue degeneration and demyelination. And it was very nice to see that with our treatment, we have less amount of pro-inflammatory cytokines in the lesion, showing that this treatment is reducing immune attack in MS, in EAE. Instead, so we have some population of actually immune cells that are good, beneficial, including T lymphocytes, which are called T regulatory or pro-regenerative macrophages. These population, we need them for resolution of inflammation and for remission. And what we found was that with NRG1 treatment, we had more number of these good immune cells, suggesting that the resolution of inflammation was promoted by NRG1 treatment. So if I want to summarize it for you, is that what we have shown is in EAE, which is the animal model of MS, NRG1 levels is severely downregulated or declined, not only in the CNS, but also in the blood. And when we treat the mouse, the EAE mouse, with NRG1 treatment, we can then reduce immune attacks. And we can have a healthy immune response. And we believe that NRG1 is a factor that promotes immune homeostasis. And when its level is declined during EAE, so then it causes disturbances in immune response and exaggerate immune response, and we have then demyelination. So then the next question was that with this treatment, can we then decrease neurological disabilities in EAE mouse? So what we did, we did a number of treatment strategy. First, we started giving treatment, NRG1 treatment at the peak of the disease. And this is when usually patients receive disease-modifying treatments in clinical management. And it was very nice to see that with a small amount of NRG1, 600 nanogram per day that we gave to the mouse, we could see a significant decrease in neurological disability. The higher number, basically higher disability. So as you see, NRG1 with 600 nanogram, this group, so they were showing less disability. Even in this heat map kind of clinical score, the more yellow that you see, it means more disability. And this is our non-treated EAE group that you see many animals, these are individual animals, that they had high disability. But with NRG1, many of them, they were really the basically severity of impairment was diminished. Then we started asking that, okay, what if we give NRG1 at the onset of the disease? This one was actually even better if we give it at the onset. Then we asked if we give it prophylactically at the time that we are even inducing EAE in the mouse during the non-symptomatic period, if we give NRG1, even some of these mice that are identified with blue, they even didn't develop EAE. So it's showing that this treatment, perhaps, it has the potential to even prevent MS if it's given even, before MS symptoms start. Even we tried post-peak that, okay, if a patient has missed the opportunity at the peak, if we give it after, so is it going to be helpful? Still, it was helpful. So overall, we have looked at different treatment strategy in mice, and it seems that this treatment has a potential. So then the next question was that, okay, so it seems that NRG1 is modulating the immune response and is decreasing immune attacks. And this is something that even several other disease-modifying treatments that are currently given to patients, recommended, in clinical management, they can also do that. So the next thing that we wanted to know is that, and this is the area that the gap that I was talking about, is if NRG1 treatment can actually promote regeneration of oligodendrocytes and remyelination. Basically, we are talking about the regenerative capacity of this treatment, in addition to its immune modulatory effects. And what we did was that we looked at generation of oligodendrocytes. So in NRG1 treated compared to non-treated, you see many red cells that are oligodendrocytes here, way more red cells, way more oligodendrocytes compared to non-treated. And also something that is important when we are generating new oligodendrocytes is their maturity. If you remember, is that, yeah, we can generate precursor cells, but these precursor cells, they need to mature, fully mature, to become myelinating oligodendrocytes. And what we found, which was very interesting, is that when we add NRG1 to oligodendrocytes that are immature oligodendrocytes, it can promote their maturation. So look at the number of processes that they are basically extending. And remember that these processes, each one of them is going to enwrap one nerve fibers. And even a study has shown that one oligodendrocytes that we can regenerate, they are able potentially to remyelinate up to 60 axons in their vicinity. So then this is a very powerful kind of, message that regeneration of oligodendrocytes is the target really for repair of MS. And it seems that this treatment has the potential. So we even looked at kind of myelination in other ways, maturity of these cells when they are kind of, they mature, they start, projecting their processes and then sheeting the axons. So then we looked at myelin itself. And these are some electron microscopy images that I don't want you kind of, necessarily to understand the entire kind of, histology here. But what I'm going to tell you is that all these profile that you see, these are myelinated axons. So what you see light is the axon, and all the black layers are myelin. And this is a normal spinal cord. We have lots of myelinated axons. In EAE, we see axons are degenerating, you see less number of axons, and you see less number of basically myelinated axons. But with NRG1 treatment, we were able to preserve some of the axons during evolution of EAE, and also we see more normally looking myelin. So we quantified that, and we found that, yes, that's the case. If this is the normal level that we see in normal spinal cord, with NRG1 treatment, we see very similar profile. While non-treated, they have less amount of myelin. Again, here, the higher number, less myelin. And also even NRG1 was able to preserve more axons. Here, higher number means more axons, which is better. So when we can preserve axons from permanent degeneration and we can have more myelin, this is a very promising then regenerative strategy. So then the question was that, okay, what's the relevance of this study that we have done in mouse for MS individuals? We always need to see if our discoveries in animal models, they can translate into basically clinical implications. So for that, we collaborated with neurologists at Winnipeg MS Clinic. They provided us with some MS samples. And we kind of confirmed some of these animal discoveries in patient samples. What we found was that very similar to EAE mouse, there is also a reduction in NRG1 level. So then the pathology that we saw in terms of decreased level of NRG1, it's actually also true in MS. So we have less amount of NRG1 in active lesions of MS. So this is an active lesion of MS. And these are all kind of the blue is the area that we have normal myelin. Blue is the myelin stain here. And then in the lesion around here, we don't have any blue staining, means that there is no myelin. And exactly when we look at the lesion area, so we have also less amount of NRG1 compared to the control area or healthy area. And interestingly, we also looked at the plasma of patients samples that we had from MS individuals, from different, basically from healthy controls, and also samples from CIS, RRMS, and also secondary progressive MS. And the data about CIS, again, I want to remind you that CIS are those individuals that have shown only one episode of MS. And normally 60% of these individuals, they convert to relapsing remitting type of MS within years after the first diagnosis as CIS. And what we found was very interesting is that CIS samples, they also showed less level of NRG1 in the plasma. And if we look at this as compared to other kind of, subtypes of MS, and if we look at this closely, so then there was a significant reduction in the plasma level of NRG1. And then we followed these patients for about, 2 to 5 years. And then when we look at these 11 patients, I know that is not a big sample, it's a small sample, but then this also population, there are not much kind of, it's a lower population of MS that we can get actually samples from. So when we looked at these 11 individuals, 6 of them, they basically converted to RRMS within few years after this kind of test. And all of the individuals that they converted to RRMS, they had lower amount of NRG1. So this telling us that it might be a potential biomarker that can predict if an individual can convert to MS or not. So other bright sides of NRG1 is that this is an FDA approved drug. It means that it has passed for other conditions, and it means that it has passed the safety in human. And then that actually facilitates translation of these discoveries after we do additional experiments. And also it has very ideal pharmacokinetic because it can passes the blood-brain barrier easily. One of the challenges for treating CNS diseases is that if we give a treatment systemically through skin injections, for example, or through intravenous, so there is always a question that if that treatment can get into the CNS because of that blood-brain barrier that I talked about. For NRG1, it can. It's demonstrated. So in addition to NRG1, there are also other advances in MS research that I'm not going to go through them, but I just wanted to mention them, like mesenchymal stem cells. They also have immune modulatory effects. Diet has a big role in MS progression and symptoms, and also exercise and physical activity. Some area of research that we are trying to fill the gaps is, again, specific disease biomarkers. We are working towards that by understanding MS pathogenesis and also uncovering what drives MS progression, why some patients, they progress quickly, is it because those precursor cells, they are not maturing in their, basically, CNS to make oligodendrocytes, approaches to protect nervous tissue from MS damage, and also strategies to promote tissue regeneration, like remyelination, and also identifying risk factors that can increase a person's susceptibility to MS, because then that can facilitate personalized type of treatment. I also wanted to mention that I'm part of the executive committee of the International Women in MS. This is my basically pleasure. With some other women, we have created, like 2-3 years ago, we have created this organization to help women who are working in MS research or patient care, to promote them in terms of the equity and in terms of their visibility within the field. So if you are interested, you can look at our website to see what activity we are doing. I also want to acknowledge my research group, funding that it has allowed us actually to do this research, and we need lots of funding. So that's, that is one of the limiting factor, kind of in our research. And also the collaborator that I have had, over the time to enable this research. Thank you, and I'm happy to answer any question that you might have.
Wonderful. Thank you so much. That was a lot of really great information. And so we do have a lot of questions. So let's bring up some of those questions if you don't mind, Chris. If you can make, or I don't know if you can make it any bigger, but let's go with this first one, which is perfect. Can you comment on MS and receiving a COVID vaccine? Any special considerations? Any vaccine better for MS patients?
Yeah, so what I recommend is that, because I'm not a clinician, what I recommend is that certainly talk to your physician, to your MS practitioner, to know about that. But as far as I know, there is no warning not to receive COVID vaccine. That benefits, because again, immune response is disturbed in MS, and there is more basically predisposition to get infection and other things, including COVID. So it's, I believe that kind of, I personally think that there is no problem, but you definitely want to talk, to your doctor to know if you should receive that or not.
Yeah. Okay. Appreciate that. That's great. You did speak near the end to this next question, which is, does diet or exercise have any impact on MS and its progression? I think you were saying that that more research is being looked at in that area.
Yes, definitely, especially in the, diet overall, having a healthy diet is good for all of us. But then again, the type of some diet, they are triggering kind of MS symptoms. So going through what is recommended, what is not recommended, that would be the best practice. Also, something that I personally think that again, you need to talk to your physician, because you don't want to remove some of the diet, some of the basically food from your diet, because they can help regeneration. For example, for making myelin, lipid biosynthesis is very important, because myelin has 80% content of myelin is lipid. So what I'm saying is that is wise to talk about, again, to your physician, and also look at lots of information that are available through the MS Society of Canada website to see which diets are recommended. And also remember that not all MS individuals are the same. MS is diverse, it's like a spectrum of disease. And it's always good to know that, okay, what type of MS you have, and then what you should do. In terms of exercise, again, in addition to the general well-being at the cellular and molecular level, what has been shown by some groups, for example, one of the group in Calgary led by Dr. Young, is that exercise definitely can modulate the immune response, and also can help production of those oligodendrocyte precursor cells that we talked about. They can basically stimulate generation of new oligodendrocytes and even remyelination. Because with exercise, certain of growth factors and trophic factors that are needed for remyelination are secreted in the body. So, and also to maintain healthy muscles, and also overall, healthy weight. This is something recommended for all of us. But
Right.
then,
Yeah.
more for MS individuals.
Does it matter the kind of exercise? Like, does it have to be an aggressive, like, how
No.
much beats per minute sort of thing? It's like a walk, enough exercise versus
Yeah.
going for a run or a swim or something like that.
No. Even yoga, for example, some traditional type of exercise, even meditate, something that it still, keep your body in an active form, and even building kind of, again, muscles, and make sure that kind of the mass, the healthy regimen that we need for nerve regeneration is then stimulated through this exercise. Walking, if and sometimes we have to consider actually the physical ability of MS individuals. Not all of them they can walk, or even some of them they might have very low ability, but even moving fingers, moving your arms, your legs, so that even can help. So, or try not to sit for a longer time, because there are lots of comorbidity associated with MS. For example, chance for diabetes, for cardiovascular. So again, the exercise, as much as the patient can handle, it's recommended. But again, there are more information available, and these are things that are tailored for each patient based on their mobility.
I think it's a good tip for all of us to keep moving, right? And as you said, the MS Society of Manitoba is a great resource. Lots of good information there. So let's bring up some other questions. So you had a nice compliment saying thank you for your presentation today. Research always gives us hope. Next question is, might the blood-brain barrier leak be primary in pathogenesis, leading to antigenic recognition of CNC structures, etc.?
Yeah. So again, we don't really know what initiates MS. Even there are different theories. Some people are saying that it's initiated outside CNS, then basically it comes kind of the immune response comes to the CNS. Now even some people are saying that maybe it's initiated inside the CNS. Again, CNS is brain and the spinal cord. We don't know, but what we know is that with MS, the blood-brain barrier is disrupted and is leaky. That's something that we know. And even there are some research going on to understand why blood-brain barrier leaks, and even how we can kind of, attenuate the destruction of blood-brain barrier. And I didn't get to present that, but what we have seen is that the NRG1 treatment, it also helps to reduce basically destruction of blood-brain barrier. And this is a treatment, actually a strategy again for MS, focusing on blood-brain barrier, but this is definitely part of the pathogenesis of MS.
Okay, great information to know, especially for those who perhaps are listening who suffer from MS, or people who they love are suffering from MS. Let's bring up some more questions. We've got a few more. Once MS has been diagnosed, is supplemental vitamin D part of the management of the disease? You did mention that earlier in your presentation.
Yeah, so I believe, again, I'm not a practitioner, but I believe that there are a number of tests that are being done. And if vitamin D deficiency is one of the basically items in those blood tests, so it's recommended. Even vitamin D, actually, it's recommended to even healthy individuals to make sure that they keep a healthy level, a physiological level of vitamin D in their body. So it's never too late to actually get supplement in terms of vitamin D. But as far as I know, vitamin D is one of the treatment strategy, and very simple kind of treatment strategy for MS. But then in terms of the dose, in terms of how long you want to take it, how often you want to take it, these are all information that primarily you need to talk to your physician, or look at those clinical forums that they have more accurate information. As I said, I'm more working on research at the cellular molecular level. Still, as one of you mentioned, research is the hope. And also remember that research actually facilitates medicine. All the discoveries that is happening, including, very first hand that we have seen COVID, those messenger RNA vaccines, those discoveries, they are all coming from research labs. So this is the part that we do, But then in terms of clinical management, in terms of, blood tests, those things, so clinicians, they have more information than us.
Great reminder. We have just a few more questions. Now, this next one, I don't have a medical background, so correct me if I'm pronouncing this wrong, but what, if any, is relationship of MS to NAION, non-enteric anterior ischemic optic neuropathy? I'm sorry. I hope you understand what helps meant.
No, actually, I'm not familiar with this clinical term, but I also know that, vision system and optic nerve is also affected by MS, even it is kind of, its own disease. But then what is exactly this term? So perhaps, no, sorry, I don't have the answer for that. That seems to be like a clinical term that you need to get accurate information, elsewhere.
Okay, yeah, no, fair enough. Two more quick questions, and that is, thanks for the information in the presentation today. I wanted to ask about the origin of neuroligin. Is it made by cells in the body?
Yes, it is. And this is actually a good thing about kind of these treatment strategies, that neuroglin-1, that's actually also a very great question. So neuroglin-1 is made by nerve cells and also those nerve fibers that we talked about. And we believe that the reason that perhaps one of the reasons that neuroglin-1 is declined is that we are losing those cells within the lesion. And this is an endogenous, like a body's protein. So it's not like other treatments that are not part of our body and we are giving it. So this is actually putting back to the body that is now declined. So, and this is how research can actually give us information about what is disturbed in MS in terms of pathogenesis of MS. So then we can work on specific targets for MS rather than just immune modulatory drugs that are given to, many of autoimmune type of diseases. So we believe that this is something that is specific to MS pathogenesis, and then again, it's not something that is foreign to the body, but then it's declined in MS lesions, and we are not trying to increase its level more than what is physiologically present in the body. We are trying to elevate its deficient levels, kind of putting back something that body has lost to make sure that then it start working more normal. Basically,
that's fascinating. Very interesting. One last question, and that is, it's with regards to acupuncture. Is it helpful in treating MS symptoms?
Yeah, so it seems that it is helpful not only for MS, for several other things, because it deals with pain and other symptoms. But then again, these are not kind of, I don't know about the cellular molecular basis of these type of treatments. And these are not definitely kind of, tackling the cause. Yeah. So these are like, equivalent to kind of, other medication that patients are receiving to alleviate pain and other symptoms. So this is a treatment that is not chemical kind of treatment, and many people are recommending that. I don't have any specific kind of opinion on that how it fits actually MS, but I believe that then it's working, alleviating pain symptoms and other things, but then it's not going to treat the cause. So it cannot be a replacement for disease modifying treatment. So that's what I want to say, is that again, talk to your patients, because some of these treatments are critical. Again, remember that MS should not remain untreated. So some sort of disease modifying treatments that your physician recommend, you need to take it. But then to alleviate symptoms, so there are other factors, even including exercise, other things. So acupuncture is one of those. That is not specific to MS, other pathologies. It can also alleviate those, but then it's not going to reduce the immune response to the point that disease modifying treatments they can. Or as far as I know, I don't think that it can, boost regeneration.
I see. Okay. Nope. That's very good advice. It does sound like seeing your doctor on some of those treatments and how to prevent some of the side effects is the way to go. So thank you so very much, Dr. Karimi. This was a really interesting, fascinating presentation. With the numbers of people in Manitoba who do suffer from MS, it's really wonderful to hear about the groundbreaking research that you're doing. I'm also delighted to see the level of donations from even alumni, from government who are supporting this research. It's really, really important that we keep learning more about MS and hopefully find a cure for it one day. So thank you for all your great work. Keep doing what you're doing. It's really wonderful, and we'll certainly invite you again to come back on what more that you have learned about the disease and whatnot. So
thank you so much. Thank you for having
You're
me.
welcome. You're welcome. And so thank you everybody for participating today. As I said, we'll send you a link. So I encourage you to view it again, as there's a lot of information that Dr. Karimi shared with us today about MS, as well as we will send you a follow-up survey to get some feedback. And I encourage you to respond, as it's the only way that we're able to improve. So next week, there is no session. We've got a week off. So the following week on June the 2nd, we will hear from Dr. Robert Mizzi, who is an associate professor with the UM's Faculty of Education, who will be speaking on LGBTQ teachers working overseas, a photo voice story. And June is actually Pride Month, so it's the topic is very well aligned in terms of what we're celebrating that month as a community. So thank you again for participating, and we will see you in a few weeks.